So really moving forward with the theme of this morning's plenaries, which is the positive emotion and reward and the new tools and techniques to examine circuitry in that area. It's my utter pleasure to introduce the third speaker of this morning, who's Dr. Michael Meany. Now, many of you will be familiar with Michael's work, but let me just tell you a little bit about him. Michael lives in two countries, Canada and Singapore. He was previously the James McGill Professor of Medicine at McGill University, and he remains the director of the Sackheim Institute for Epigenetics and Psychobiology at McGill. But he is now also a senior investigator in the Agency for Science and Technology and Research in Singapore and leads the Translational Neuroscience Program at the Singapore Institute for Clinical Sciences. Now, he has had so many awards, it's going to be absolutely impossible to tell you about them all, but I'm just going to give you some edited highlights. He won the Wilder Penfield Award in 2014, which is the highest award for biomedical research given by the government of Quebec. And in the same year, he won the Klaus J. Jacob Research Prize Laureate, which is one of two international prizes presented to honor scientific achievements of exceptional social relevance. He was also Radio Canada Scientist of the Year in 2009, so he's obviously a very engaging speaker. And he's a member of the Order of Canada and the Knight of the National Order of Quebec. As many of you will know, Dr. Meany's research focuses on very elegant work, focuses on the epigenetic mechanisms for the effects of early experience on gene expression and neurodevelopment. And today he's really going to give us a slice of a slightly different aspect of his work, which is the role of maternal mental health. And he's going to talk about positive maternal mental health, reframing the discussion of maternal mental health. So it's my delight to welcome Michael Meany.
Thank you so much. Thank you, Mary. And I'm going to apologize for the confusion on the titles. The confusion, which those of you who know me isn't totally out of character, referred to the actual invitation where I thought it was plenary, but I've changed the topic, and so although it's something quite different from what you've heard previously, it is one that I think thematically is much more closely aligned to a wonderful idea of focusing on positive emotion. So as Mary suggested, I'm an individual with lots of affiliations, but to the best of my knowledge, no conflicts, at least with respect to this particular topic. So I spent a great deal of my career, and our group continues, to try to understand maternal influences over gene expression and brain development. And we've used model systems, classically in the rat, more recently in mouse. And in the last while, I've had, and particularly with the context of our work in Singapore, the opportunity to translate that work into human studies of child brain development and function. And I'd kind of like to walk you through some of the impressions of an individual moving from basic neuroscience and looking at social modulation of gene expression and brain function into trying to adapt those studies in humans. And of course, I think the primary place one begins in looking at maternal influence in the context of human studies is looking at the impact of maternal mental health. What you're looking at right now is a study published by the London School of Economics that has analyzed the impact on the British health system of maternal mental health problems. And two things are worth noting here. First is the magnitude of the impact. It is $8.1 billion per year. And for those of you who work in the area of pediatrics, we're not simply talking about what we would think of as childhood problems associated with behavior, cognition, or emotion. We're talking about the rudimentary problems that pediatricians see every day, such as asthma, recurrent abdominal pain, etc. All of these are influenced by maternal mental health issues. And the second point I think that's really prevalent on this particular slide is to note that almost 3/4 of the costs associated with maternal mental health are related to treatments dedicated in targeting the child. So clearly we're looking at an influence that is pervasive. So the translational aspect and opportunity emerged through the emergence of a longitudinal birth cohort study we established in Singapore in 2009 due to the funding either through A-STAR core funding or from the National Medical Research Council in Singapore. We began by recruiting in the first trimester of pregnancy about 1,100 women who are of the major ethnic groups within Singapore, including Han Chinese, Malay, and Indian. This study features heavily and deep phenotyping of both the mothers and the children. And so what you're looking at here are the number and the frequency of laboratory-based visits assessing mother and child. And the primary focus here is to really understand the developmental origins of individual differences in vulnerability for either metabolic disorders, childhood obesity and pre-diabetic states, or for mental disorders. The study is also unique in the sense that it involves the largest attempt to biologically phenotype both mothers and children. And so the study involves genetics, epigenetics, transcriptomics, proteomics, metabolomics, which will feature towards the end of this talk, as well as the microbiome. And you really haven't lived until you've turned to the mother of a 2-week-old child and asked her to collect the fecal material of her child in a Ziploc bag and store it in her freezer. The things of which translational science are made. Okay, so let's start with really the simplest point here is we're obviously characterizing the mental health, the quality of mental health in the mothers in the study. You're looking at what is one of the 2 primary measures used to assess symptoms of depression. This is the Edinburgh Postnatal Depression Scale, which, by the way, is valid for prenatal states as well. The CES-D from the CDC in this country would be the other major tool. You're looking at the distribution. This is the frequency of depressive symptoms on the ordinate. You're looking at the frequency, population frequency of individuals who score within, first of all, the range of probable clinical depression. And on the EPDS, that includes individuals scoring 14 or 15 and above. So in the Singapore cohort, 10% roughly of the mothers score within that particular clinical range. That's almost exactly the number that you'd find in the United States, in Australia, in Britain, in Canada, etc. So the distribution is very similar, skewed to the left. What you also will note, however, is a very substantial portion of mothers that fall into the high subclinical range. Almost 30% of the women fall within that range, and that's going to be a major focus of the next few moments of this talk. But this is just generally the distribution. The other feature I think that's really quite interesting to keep in mind is that although traditionally we've used the term postnatal depression, it's a misnomer. The vast majority of cases, either clinical or in the high subclinical range, have a prenatal onset, normally within the first trimester. In fact, you can even see evidence preconceptually of these particular conditions. Unique postnatal onset is rather uncommon. It does occur, but it accounts for about 1 to 2% of the cases. And so most consistently, what you see is that the symptoms either of depression or of anxiety are established in the first trimester and largely persistent into and throughout the postnatal period. Now, the second major point to be made here is that the influence, the associations that one sees between antenatal maternal mental health or postnatal maternal mental health and various child neurodevelopmental outcomes cuts across the population. It is not unique only to those mothers with clinical levels of symptomatology. So recall for a moment the actual cutoff here is about 14 or 15 for women with clinical levels of the EPDS. This is a summary of studies from 3 different cohorts looking at multiple outcome measures. The top A and B are neuroimaging studies. C is from the Generation Rotterdam study and looks at internalizing and externalizing problems in childhood. What you see from our own Gusto study in panel A is that there is a linear relationship between the EPDS symptoms and amygdala microstructure. It cuts across the population and is not unique to only those individuals within the clinical range. And this, in fact, is even more striking in data on cortical thickness from the University of California Irvine cohort, in which again you see a negative relationship between cortical thickness and symptoms of depression on the EPDS, with only 1, maybe 2 mothers in the clinical range. Again, it cuts across the population, and that theme is, I think, shown compellingly in the Generation Rotterdam study. This is symptoms of maternal depression and looking at both internalizing and externalizing childhood problems at 4 years of age. You can see that for mothers with no, low, moderate, which would be high, and clinical levels, that there is a graded relationship between the symptoms of depression and the severity of either internalizing or externalizing problems. So the simple point here is that maternal mental health issues cut across the population with respect to child outcomes, and there is also compelling evidence that the same commentary can be made in relation to the mother's own psychosocial functioning. There are a range of studies which have shown that impairments in psychosocial function are, in fact, comparable between women with clinical levels of depressive symptoms and women with high subclinical levels of depressive symptoms. So the bottom line here is that, in fact, it is not simply these women with the clinical levels of depression in which one sees outcomes in relation to either child development or the mother's own psychosocial functioning, that includes at least those women with both clinical as well as high subclinical, which actually accounts for as much as 40% of the population. And what the evidence does indeed suggest that 40% of pregnant women show symptoms of depression sufficient to affect their own functioning, as well as that potentially of the neurodevelopment of their offspring. So 2 themes I want to try to explore here that are interrelated, and the first is one I think that sort of sets the stage for the second, is that while we've often referred to these circumstances as referring to the postnatal period, that in fact there is good evidence that the major effects may actually be operating during the antenatal phase in utero. So first, some evidence for the importance of maternal mental health in mediating the impact of more proximal variables. So not surprisingly, mothers in low-income families show a higher degree of symptoms of both depression and anxiety during both the antenatal and postnatal phase. And what you're looking at here is just the increase in the frequency of symptoms of women in the bottom quartile versus women in the top quartile. And again, this comes from the Gusto cohort study, but it could have been just about any study. This is well established within the literature. The point, I think, that underscores the importance of maternal mental health is that a range of studies, including those done in this country as well as our own studies in Singapore, show that maternal mental health either partially or completely mediates the association between socioeconomic status and child developmental outcomes. And that refers to both socio-emotional outcomes as well as cognitive outcomes such as executive functions and functional outcomes such as school readiness. What has emerged from the Gusto study, uniquely, that was really quite surprising, is that if you look at the ability of maternal mental health to mediate the impact of socioeconomic status on these outcomes, there is stronger evidence for antenatal maternal mental health. And that is true not only for things like socio-emotional problems, even for outcomes such as school readiness and executive functions. You see evidence for postnatal mediation, but the evidence is stronger for antenatal mediation. And that's true even for outcomes that are assessed at 4 years of age. Now that is, of course, somewhat surprising, but it is also well aligned to studies from the Avon longitudinal study of parents and children. Rebecca Pearson and her colleagues have followed up on the well-known finding that children of depressed mothers are more likely to experience depression themselves. As it turns out, that association between depression in the mother and the likelihood of depression in the offspring is stronger for the antenatal depressive symptoms of the mother than it is for postnatal depressive symptoms. And another finding that aligns with those in the cognitive field from the same cohort is the work of Jonathan Evans and colleagues, showing that the antenatal, but not the postnatal period, is actually a sensitive period for the effects of maternal depressive on cognitive development, expressed in this case as a issue of IQ. So the epidemiological literature would suggest that maternal mental health is shaping brain development in a way that influences later function in both the socio-emotional as well as in the cognitive sphere. There were studies well before we began with Gusto suggesting that these effects do have a structural and neural basis. The work, for example, at the Irvine group, amongst others, had shown that fetal exposure to maternal depressive symptoms alter associates with cortical thickness in later childhood. Work by Sonia Lupia and others had suggested comparable effects in the amygdala of the hippocampus. The problem with these studies, however compelling, was that in each case, the imaging had actually been performed in late childhood. And so we took advantage of the cohort study in Singapore, as well as with the expertise of neuroradiologists such as Myriad Fortier, to design the protocols by which we could image children shortly after birth using, of course, unanesthetized preparations. So this is imaging of kids within the first 10 days after birth. This is a community-based sample. It's the Gusto sample, and allows us to more directly assess then the associations between maternal mental health in the antenatal phase and brain structure and connectivity. This involved, of course, the work of Chuan-Ji to develop infant brain atlases for the sake of analysis. And just as a demonstrate, this work has been published some time ago, but essentially what one sees is that neural circuitry that we commonly associate with mood disorders, such as connectivity between the amygdala and hippocampus and, of course, the prefrontal cortex, is strongly influenced by antenatal maternal mental health. And I'm just going to give you a little bit, a few examples of this with respect primarily to the amygdala. This is work by Anne Rifkin-Graboy in the Gusto cohort, showing the association between fractional anestropy of diffusion tensor imaging and the symptoms of depression as measured on the EPDS. As you can see from the previous graph, this cuts across the entire population. And the effect, at least with respect to symptoms of depression, is highly specific to the right amygdala. The left amygdala is unaffected. Chuan-Ji in her laboratory then did work on connectivity, and what they showed was increased connectivity between the right amygdala and a number of prefrontal regions as a function of maternal symptoms of depression. The most interesting finding, I think, in this is that of greater connectivity between the right amygdala and the insula, which is actually a hallmark of individuals who suffer from anxiety disorders, suggesting potentially that the patterns of connectivity that associate with vulnerability for mood disorders may actually be established over the course of antenatal development and in association with the quality of maternal mental health. Importantly, these structural effects observed at birth are predictive of later behavioral outcomes. This is simply looking at internalizing symptoms on the ordinate as a function of the right amygdala structure at 24 months of age, and you see a significant relationship. That relationship does not statistically persist into 48 months. What's interesting is what emerges in contrast is a relationship with the insula, suggesting that that pattern of connectivity is associated with later behavioral development. And that pattern has also been seen in the work of Damien Fair and Claudia Bus and their colleagues, again looking at the same measure, that is amygdala connectivity at 6 months of age, and it is predictive of fear and cognitive development at later points in time. So importantly, what is established over the course of antenatal development appears to be predictive, at least over periods of early childhood. And I mentioned the amygdala, but there are papers, for example, coming from our group and now other groups, particularly that of Claudia Bus, looking at associations not only with the amygdala, but also with the hippocampus and prefrontal morphology. In many cases, these effects are moderated by the genotype of the infant, but they do speak to the rather pervasive influence of maternal mental health on this particular circuitry. Okay, this was essentially intended to convince you that the antenatal period does hold enormous importance in terms of the associations between a maternal mental health, child neurodevelopment, and outcomes. The second point that speaks more to the theme of this particular seminar is, in fact, that of looking at maternal mental health along a continuum, and rather than simply being focused on symptoms of depression or anxiety, of considering the quality of maternal mental health in a broader perspective. And of course, I think this simply reminds us of what we all know, is that the quality of mental health should not be defined simply by the presence or absence of symptoms. And that has been the work of Desiree Fuao, a postdoc in Singapore, who took this on with a passion. So Desiree took the data that we had been collecting from others at various points in time. You can see that in the context of Gusto, that involves prenatal and multiple postnatal sampling. She took the individual items of the Edinburgh Postnatal Depression Scale, the State Trait Anxiety Inventory, and the Beck Depression Inventory. And she did bi-factor modeling to look at the individual items and how they cluster. And not surprisingly, what she found was that there was a general factor, and basically all the symptoms load on this general factor, and those include symptoms of depression and anxiety. And of course, there's nothing surprising about that. These are commonly comorbid conditions. But what the general factor does allow you nicely to do is to integrate the conditions of anxiety and depression into a single measure of maternal mental health, which is important because, of course, in the maternal field, as in others, the comorbid conditions of depression and anxiety represent more severe conditions. But the more interesting component of this was that the same bi-factor modeling allowed her to identify positive mood and positive self factors from items, largely those on the STI. And I apologize that this is so ridiculously small, but I'll just give you an idea. These are all fairly intuitive. Positive mood, for example, is characterized by women when they endorse items such as I feel calm, I feel self-confident, I'm relaxed, I'm satisfied with myself, I feel secure, I'm a steady person, I'm happy, etc. And all of those items can then be collected into a unified factor that represents the quality of positive maternal mental health. And so the remainder of the talk is really meant to convince you that this is an important consideration. Unless you think, and this would be perfectly understandable, that positive mental health is simply the flip side of negative mental health, I can persuade you otherwise. First of all, as you'll soon see, they have very different consequences in terms of child development. Second, the level of correlation or anti-correlation between positive and negative mental health is not nearly as strong as you would think. It's about 0.25. Right? So clearly there is some association, but it's not by any means simply the reverse of negative mental health. They have different antecedent conditions. Different factors predict positive and negative mental health. And so these really are two very different dimensions. Another way you can see this particular network is that Desiree has also been doing symptom network analysis. This is again from the State Trait Anxiety Inventory in both the prenatal and in the postnatal, and although this is absolutely illegible, what you can see is that in blue are items that represent the positive mental health component. In yellow are items associated with anxiety, and you can see that these are very distinct symptom networks and that they persist and are largely stable between the pre and postnatal phase. And again, it endorses the idea that there is a very distinct network of features related to positive maternal mental health. I don't think any of that's terribly surprising. What is a bit surprising is that the topic actually, although it's been almost completely ignored with respect to child development, there are a number of studies that have actually shown that positive maternal mental health associates with better birth outcomes, which is rather interesting because the evidence here is rather compelling. In contrast, meta-analysis show that symptoms of depression and anxiety actually are not really great predictors of birth outcomes, either by looking at gestational age or at birth weight. The studies on positive mental health are rather consistent, particularly in showing a reduction in the risk for prematurity. And so I'm sort of giving away a little bit of where I'm going here, but do note, for example, as I'm sure many of you know, that prematurity is a condition, a maternal condition, that reflects enhanced inflammatory activity. So with this in mind, we decided to start looking at the specific associations of the general factor, again, reflecting symptoms of depression and anxiety, and the two positive factors, maternal mood, positive mood and positive self. What you're looking at here is the type of data one collects over many years within a longitudinal cohort study. These are measures of social and executive function, social problems, the Q-chat, of course, which is a screening tool for autism, various health measures, and of course the Baileys. All of this is meant to show you on this heat map that it is not at all hard to find outcomes, neurodevelopmental outcomes in children, that are statistically associated with positive mood, but not the negative mood of mothers. Conversely, of course, especially in the health sphere, as well as in many measures of socio-emotional outcomes, you find outcomes that associate with the negative symptoms, anxiety and depression, but not with the positive. So the outcomes are distinguishable with respect to, in this particular case, positive and maternal and negative maternal mental health. And you can see that at the level of structure to some extent as well, although we've just begun to really pursue this particular topic. Recall in the previous study of Anne Rifkin, there was this negative association between symptoms of depression and a microstructure in the right amygdala. Don Koh's analysis have shown that if you look at the positive, for example, what you see is exactly the opposite effect, that in fact, looking again at diffusion-weighted tensor imaging analysis, that there is an increase in microstructure in mothers high in positive, in the left and right amygdala in the offspring, with generally only a trend associated with the general. So the positive, thus, has statistically distinguishable effects that one can see at the level of neurodevelopment. And this outcome, again, has behavioral implications, that when Dawn followed these children through and looked at behavioral dysregulation at 12 months of age, she was able to show an association between amygdala microstructure in the left amygdala and behavioral dysregulation. There was a trend, but not significant, in the right amygdala, again suggesting that these effects portend to later outcomes in these children. And then we ended up with another surprise, because Evelyn Law, who had been leading the studies, looking at maternal mediation of the effects of socioeconomic status, she then analyzed these outcomes, looking at socio-emotional problems as well as cognitive function, what included global measures such as IQ, specific measures of executive function, or outcomes such as school readiness. And what she found in terms of her model with antenatal mediation is that for each of these cognitive outcomes outlined in red, it was the positive but not the general or negative factor that was the mediator. For the socio-emotional problems, it was in fact maternal symptoms of anxiety and depression, but for these more cognitive associations, it was the positive maternal mental health that mediated this association. We've then started to look, and this is the work of Michelle Key, which is actually summarized in a poster that will be presented tonight. Michelle's poster title and poster will obviously give far more detail. I'm simply going to summarize this work. But Michelle has started to extend these analyses into the postnatal sphere, and what she's done is to look at the associations between positive and negative maternal mental health and various forms of parental styles. These include forms of parenting and parenting styles that enhance the probability of child psychopathology. Those are permissive and authoritarian parenting styles, as well as a form of parenting, authoritative parenting, that diminishes the probability of child psychopathology. What Michelle finds, I think not surprisingly, is that looking at the general factor, which again captures that integrated measure of anxiety and depression, is that it is positively associated with both permissive and authoritarian parenting and negatively associated with authoritative parenting. Positive maternal mental health has a very unique impact. It is strongly associated, but only with authoritative parenting. And what Michelle's poster will take you through is not only this, but also its association with maternal childhood adversity. She'll also show you mediational data. In essence, I'm just going to briefly summarize this, that these parenting styles do show mediational effects, so that the association between negative maternal mental health and multiple child outcomes of socio-emotional, including externalizing and total problems on the child behavioral checklist, is mediated by permissive parenting as well as authoritarian parenting. Authoritarian parenting also at least partially mediates some outputs in the cognitive functioning, looking at school readiness as well as IQ. Authoritative parenting in the positive is pervasively associated as a mediator with both socio-emotional outcomes as a whole, as well as a whole range of cognitive outcomes, including executive functions such as working memory. So these features of child developmental outcomes then have specific antecedents associated with both negative as well as positive mental health, and that extends into the postnatal period. So lastly, surprisingly, despite the evidence and for the importance of maternal mental health and childhood outcomes, there are surprisingly few studies looking at the actual biology of maternal mental health in the antenatal or the postnatal phase. And so we decided to start looking at this, taking advantage of the biological samples that exist within our Gusto cohort. And I won't go through all the reasons for this, but the reason we focused primarily on metabolic data, and particularly in a lipidomic structure, is previous evidence that had associated the quality of maternal mental health and in particular symptoms of depression with lipids, and notably with specific free fatty acids. For the most part, those epidemiological studies involved dietary reports and then assumed levels of free fatty acids in plasma. And instead, what we decided to do is to actually use a targeted lipidomic analysis looking at over 450 lipid species using liquid chromatography mass spectrometry. So what we're doing here is we've collected conceptual tissue in the context of the Gusto cohort. We've obviously characterized maternal mental health in the antenatal sphere, and we, of course, have developmental outcomes in the child. And what we found was that looking either at the maternal or the fetal compartments of the placenta, there are lower levels of phospholipids containing long-chain polyunsaturated fatty acids, notably DPA and EPA, and higher levels of arachidonic acid, and that profile associated with higher levels of maternal depressive symptoms. Now, that's in the placenta, which of course captures the window at the end of the period of pregnancy. So we asked the question as to whether this lipid signature existed prior to birth and over the course of pregnancy. In this particular case, we looked at lipid signatures in maternal plasma. And again, what we found is that particularly free fatty acids associated with phosphatidylcholine, notably DPA and EPA, but not DHA, were significantly associated with maternal depressive symptoms. So there is a lipidomic signature that is strongly associated with maternal mental health. And what I want to do, just to underscore the strength of the specificity of this effect, is to actually just run through the actual raw statistical analysis that you see. So what we're looking at here are the actual lipid species that associated with the negative score. That's the score that reflects the integrated levels of depression and anxiety. And you can see there are a number of species, sphingomyelins in particular, that are associated significantly with maternal negative symptoms and that pass false detection ratios. But that's for the general. This is for the positive. And you can see that the statistical strength of the association between these lipids and the positive measures of maternal mental health overwhelms that for the general. There is little trouble finding multiple species, ceramides, sphingomyelins, etc., that easily pass false detection rate. And indeed, the strength with this is much stronger with positive compared to the general. Now the question then becomes as to whether this is really meaningful in the overall schema, and one of the ways we've tried to look at that is to ask whether or not these pathways are predictive of later child outcomes. So in the context of the work by Nirja Karani, who is one of the collaborator in the context of the lipidomic analysis, okay, now I'm experiencing the same issues that the previous speaker did, is that if you look at maternal levels of DPA, EPA, either in the placenta or in blood, that you can then break those down into specific species, notably phosphatidylcholanes, right, containing these long-chain free fatty acids. And each of the species that you see right here statistically mediates the association between antenatal maternal mood and multiple childhood outcomes at 4 years of age. And that includes socio-emotional function as well as ADHD-like phenotypes, notably inattention. So these lipid species then are actually statistically mediating the relationship between antenatal maternal mood and these particular outcomes. And keep in mind that for the most part, these lipids are more strongly associated with positive maternal mental health than the negative components. Don Coe has gone back over some of our neonatal imaging, again to try to look at the biological plausibility of these particular hypotheses. Don took the neonatal structural data, for example, of which we have about a little less than 200 children imaged, and she simply asked the question, what are the factors that we've measured of the maternal phenotypes that best predict inter-individual variation in multiple brain structures? When she looks at the amygdala, for example, she sees what we've seen before in more targeted analysis, that maternal mental health is in fact a major predictor. If you look at the hippocampus, however, what you find here is that the primary predictor of inter-individual variation in hippocampal morphology are these omega-3 fatty acids, and in particular the relationship or the ratio between omega-6 and omega-3. And you can also see evidence of this omega-6 influence down in the cottage in the midbrain. Dawn then took the various species. She did a cluster analysis and a hierarchical analysis of the various lipid species, categorizing them into omega-3 and omega-6 fatty acids. And this started to give us some insights into what might be an operative biological process. If you look, for example, at the cerebellum, at the hippocampus, at gray matter, white matter, in general, what you see is that with these omega-3s, and particularly DPA and EPA, you generally see a positive relationship between structure and the levels of these particular lipids. If you look at the omega-6 in general, you see exactly the reverse. And of course, the interesting feature of this is that omega-3 fatty acids are anti-inflammatory, omega-6 fatty acids are pro-inflammatory. And in fact, one of the major features here is that it's the relationship or the ratio of omega-6 to omega-3 that was in fact signature of this particular difference. And this is entirely consistent with a wonderful paper published last year, again from Damien Fair and Claudia Bus's group, looking at the relationship between maternal cytokine levels during pregnancy and newborn brain connectivity, and then later working memory and identifying IL-6 as a highly influential factor. Another piece of evidence, again, supporting, if you will, the implication of inflammatory processes is that when our group collaborated with Nirja Karani to look at genetic variants that moderate the association between antenatal lipids and maternal symptoms of depression and anxiety, we found a significant association between a variant in the TGF-beta3 receptor, which of course mediates pro-inflammatory signaling, and this particular relationship between the lipids and maternal negative mental health. And again, what that suggests, again, is just simply implicating inflammatory processes within this process. And for those of you interested in a more systematic and intelligent review of this particular topic, there's a recent paper in Biological Psychiatry summarizing the evidence from both preclinical as well as clinical studies implicating inflammation as a proposed mediator between maternal stress and offspring neurodevelopment. So in summary, I hope we've walked through something I think that is compelling with respect to evidence into areas that are clearly much more speculative. Lying within the sphere of those that are somewhat more compelling, I think, is the evidence that suggests that, first of all, the influence of maternal mental health cuts across the population. We have, of course, with good reason, focused clinical efforts on women with clear signs of depressive or serious anxiety disorders during pregnancy. We need to recommend that this entire consideration be expanded to a much broader extent of the population. Literally, as many as 40% of all women are affected by symptoms of depression or anxiety to a level that will affect their own health and well-being as well as that potentially of their child. Potential influences, obviously, again, simply a message many of you which are familiar, is that mental health is bidimensional. There is, of course, a positive dimension, and the positive dimension in mental health, as is emerging, I think, within many fields, has very distinct antecedent conditions, and at least in terms of child development, it has very distinct influences, both in terms of the antenatal and the postnatal outcomes. There may, in fact, it seems, be a specific signature associated with this that includes such things as free fatty acids. And these are, I think, ideas worth pursuing, however speculative, because, of course, one of the problems that we run up against in the treatment of women, particularly with antenatal depressive symptoms or those of anxiety, is an obvious reluctance to move towards medication. If we can work towards such alternatives, I think they become more broadly distributable. They've been taken for years, and of course the safety and security of these products is well known. And finally is a hypothesis. It's consistent with many preclinical studies looking at free fatty acids, but is the hypothesis that these effects may in fact involve some sort of a buffering against the hyperinflammation. Again, what's interesting is to recall the associations with gestational age, a condition of hyperinflammation, is the idea that these nutrients may in fact be targeting what many people think of as an operative pathway towards symptoms of depression and anxiety. And thank you very much for your patience.
We have time for a question or two, and I want to commend Dr. Meany for getting us back on schedule. So that positive emotion flexibility.
Joe Hibbelin here. Fabulous talk, great science. I only want to underscore a couple of points. One, first, that these are essential fatty acids that cannot be made de novo, and that they selectively get concentrated in the synapses and make up 20 to 30% of the membranes of the brain. So mothers' and babies' brains compositions depend almost entirely on how much seafood mothers eat, the source of the omega-3s, in comparison to fried foods and omega-6 fatty acids. The question, so it's possible that the low socioeconomic status driving away from high-quality foods and into poor-quality foods may be mediated also through diet.
Yep. Joe, thank you, first of all, because your first comment, for example, is a slide that should have been shown in the context of this talk. You're absolutely right. These are essential. And so, for example, one of the things you find if you look at maternal plasma levels and fetal placental levels is the correlation is about 0.95, with the 5 being noise for the most part. And you're absolutely right in terms of membrane development and how incredibly important these are for synaptic membranes. The issue of diet's really intriguing. Historically, for example, people who know far more about this topic than I do point out that if you look at hunter-gatherer diets, or historically diets, there was about a 1-to-1 relationship between omega-6s and omega-3s, and that relationship is now skewed heavily in favor of omega-6s. So you're absolutely right about historically, and I think the comment on the socioeconomic component is absolutely well worth pursuing. And you just gave us a really good idea. You're spot on. There's no reason we couldn't look at that particular feature. And of course, in Singapore, as in other places, the dietary plasma levels are associated with socioeconomic status. Yeah, thank you very much.
Sorry, we'll have to move we'll have to move on to the next speaker. Sorry, Suzanne. We're on a tight schedule.